differentiation. Today we're doing 3.5 cell growth and division. Cell growth and division. There's a somatic cell. A somatic cell is every cell in the human body except the ones that produce sperm and eggs. A homologous pair is a pair of chromosomes in the two copies of a single chromosome found in each somatic cell, making a sister chromate. A diploid is a pair of chromosomes found in each somatic cell of the body. Interphase is a period of the cell during which the cell is not divided and includes the G1 phase, growth phase, in a cycle, and an S phase, in a phase where DNA is replicated. Meiosis, the division of genetic material during which the cell nucleus breaks down into new fully functional nuclei are formed. Cytokinesis divides the cytoplasm into two distinctive cells. So it's basically a process where a cell is replicating. A sister chromate, each copy of the chromosome is referred to as sister chromate and is physically bonded to another copy. So as you can see, these two are sister chromates. Centromere. The centromere is a structure that attaches one sister chromate to another. This is because a human has 46 chromosomes during this phase. There are 92 chromates, 46 times 2 in a cell. As you can see, it's right here, centromere.
To begin, I'm going to go slides. That's my wife as my home screen, by the way. Me and Carolina, we actually did the differentiation slide together. So, I'm going to do the first two bullet points. So, to start with that, cell differentiation is how generic embryonic cells become more specialized cells. So, it changes shape, size, and its function, and how it the process of it starts with the fertilization of the female egg. So as soon as the egg is fertilized, the cell multiplicates, and for it to also differentiate, it also has to manipulate the genes. um we put the two pictures down there, and we also cited it I'm sorry, um like you asked. So yeah, bye.
So, a transcription factor is one of a class proteins that bind to specific genes on the DNA molecule. So, when it says specific genes, it could be referring to genes that are being passed down from your family, such as your parents or your siblings, or any family member in general. It's as simple as that. Then, the primary mechanism by which genes are turned on or off is by transcription factors. And if you look at the picture below, you can't really see the wording or the letters on it, but the totoponent embryonic cell is on the very top, and that is being pointed down to the pluripotent embryonic stem cells, which are the endoderm line, mesoderm line, and the ectoderm line. Then those are being pointed down to the multiponent stem cells, and those on the bottom are being pointed down to the lung, pancreas, heart muscle, red blood cell, and skin and neuron.
Well, alright, so it's come my time to do my slide, which is 3.6, and it is titled Stem Cell. What a stem cell is an unspecialized cell that can divide itself without limit. Under that, we have totipotent. Yeah, the first, which is the first embryonic cell that arises from the division of the zygote, are the ultimate stem cells. Next, what we have next is pluripotent, which is a stem cell and is one of them that has the most potential to differentiate into any type of human tissue, but cannot support the full development of an organ. Next on the list, we have multipotent, which is a stem cell that has the potential to differentiate into different types of cells within a given cell lineage or small number of lineages, such as red blood cells or the white blood cells. Next, we have oligopotent, and it is limited to become one of the few different types of cells. And lastly, what we have on this list is unipotent, and it is fully specialized and can only reproduce to generate more of its own specific cell types. And on this next slide, you can see I have a picture of what a stem cell actually is. And this stem cell, for example, is composed of the muscle cells, blood cells, liver cells, and intestinal cells. Yeah. And that concludes it for my slide. On to the next.